Hello, everybody, and welcome to what I think is going to be a really exciting session on global resources to support biodiversity genomics. We've got a very crowded session today, so I'd like to move straight on to introduce our first speaker, my colleague Bill Baker from the Royal Botanic Gardens, Kew, a celebrated palmologist, which doesn't mean he'll tell you a fortune, but means that he's an expert on the Ericaceae. He's going to talk to us, I think, about the Plant and Fungal Trees of Life project.
Well, hello everybody from London. My name is Bill Baker. I am Head of Comparative Plant and Fungal Biology at Kew Gardens, and I also lead the Plant and Fungal Trees of Life programme at Kew, and that's what I'll be talking to you about today. But first of all, I want to credit the team. This is a very brief high-level summary of an enormous amount of work done by a very large team of people, in fact, not all of them captured here. So I'd like to flag their investment and their hard work for the project, which has made it what it is today. So I'm going to start with a pretty big question, I guess. What do we know about the plant tree of life? Well, it turns out we know quite a lot, actually. The plant community has been very effective at collaborating over the years. And we have, for example, some very, very big trees available at the moment. This tree on the left, the Smith and Brown tree of 2018, it contains the majority of all vascular plant species in it. But what you'll notice about it is that a lot of the tips are blue, and blue indicates that there are no public DNA data available for those tips, for those species. And in fact, those tips have been interpolated into the tree using taxonomic surrogates. And we know, in fact, that perhaps only about a quarter of all vascular plant species have any data at all in public DNA repositories. So big trees, but problems with the data underneath them. And then on the right, we have a fantastic example of a big data phylogenomics project published just last year by the 1000 Plants, the 1KP initiative, showing coverage across the green plants this time, over 1,100 species. A tree built from transcriptomes, pulling out 410 single copy gene families. An amazing piece of work, really benchmark piece of transcriptomics for plants. And yet, of course, compared to the big tree on the left, the species sampling is minute. So we have this tension: big trees with issues with the data underneath them, and big data trees with issues around species sampling. And of course, we're all hoping that the big global biodiversity genomic agenda is going to set that straight. We have the visionary moonshot project of the Earth Biogenome Project we're talking about so much at this meeting. And this project is having really amazing impact. The Darwin Tree of Life being one piece of such impact. But of course, we have to recognize that there are significant limitations to achieving the objectives of the Earth Biogenome Project. Achieving taxonomic scale and sourcing samples top quality DNA is a real challenge for these agendas. So what would we do to address that? To what kind of place would you turn? And I hope by now all of you are convinced that the collections-based institutions are really the place to look for help. A place like Kew has amazing living collections, perhaps some 40,000 species, has fantastic connections around the world, partnerships in 100 countries rather. You can see some field work going on in New Guinea. Fantastic facilities. They're great places to really drill down into biodiversity genomics. And in fact, we've looked across the flowering plant genera at Kew, looking at our kind of ideal genomic tissues in our living collections, in our seed bank, in our DNA bank and our tissue bank. And we find that for the flowering plant genera, we have 57% of them already available to us in good quality tissues for genomic purposes. But there's still a big chunk that are missing. And what would we do about that chunk? Well, how about going to the herbarium, where we have more plant species than far more than in the living collections? There's now a large literature showing quite how much we can do with the herbarium to obtain DNA. Now, of course, that DNA is not of the same quality as you would get from a living plant. So to work with these specimens in a genomic context requires some compromise, which is why for many of us working with the big herbariomic push, we're now focusing on targeted sequence capture as the optimal approach, a method that can pull out thousands of genes from even the most fragmented of DNA. And that's really the context for the Plant and Fungal Trees of Life project at Kew, which aims to complete the plant and fungal trees of life for all genera. Now, of course, what does complete mean? For us, complete is one species per genus. By cutting it down like that, it means I can sleep at night. And in the case of the plants, we focused specifically on the angiosperms, where we have real strength. Now, that's about 14,000 genera. And in pushing through the angiosperms, we really want to exploit our collections and prioritize particularly on data gaps, on taxa that do not have any data in the public repositories at all as yet. And to deliver this, we decided to create a kind of a universal toolkit. The plant community have been very effective at kind of building in the potential of the data that is gathered by really focusing on a core set of loci during the Sanger sequencing days. And yet, as the phylogenomic potential of target capture and other approaches, really hits home, we could end up sequencing all the way across all possible gene opportunities, and then have real difficulties pulling the data together for synthetic purposes in the future. So, as part of the PAFTOL program, we have created the Angiosperms 353 probe set, which is a universal toolkit that will pull out 353 loci across all the angiosperm families, all 400 plus angiosperm families. It's even effective at a wide range of taxonomic scales. We put all the data underpinning that, the probe sequences, out there in the public domain, and it's widely accessible, it's cheap to use, and it's published in a paper in Systematic Biology last year, and you can follow the GitHub for more details. How's it worked? Well, it's been amazingly effective. You can see from these charts of genes retrieved above 50% of the target length that we're getting a lot of data out of a wide diversity of material. Now, there is a bit of a slew across the y-axis there, which you would expect from a universal kit, but the majority of data points are squished up in that top end of the graph. What you can also see by the color coding there is that herbarium material, even old herbarium material, is remarkably effective, and even specimens up to 100 years old are producing large amounts of high quality data, almost as much data as you could expect from a really good quality silica gel dried specimen collected only recently. So this really works. And we've got quite a long way so far, before COVID interrupted, of course, but we've sequenced around 5,500 samples as yet. That covers around 4,500 genera of the angiosperms. That's about 32% of the total. And we hope that by the middle of next year, we will have hit sampling of 50% of the genera in every angiosperm family. So that's what we're aiming for right now. We are collaborating very widely. There are more than 50 projects linked with our program at the moment, working across taxonomic scales. We're seeing wide uptake across the community, people who are working with this kit without any connection to our program at all, which is fantastic. For example, we've got this fantastic phylogeny of the tropical Asian pitcher plants, Nepenthes, right down to the species level, using 353. And on the right there, you can see C4 sedges, really important, for as ecosystem engineers in certain parts of the tropics. Difficult radiations that have been now unpicked using the Angiosperm 353 toolkit. We've also seen other big programs take up the kit. The Genomics for Australian Plants project, for example, aims to sequence all the Australian angiosperm genera, and they have taken up the Angiosperm 353 kit and are coordinating their sampling with us in PAFTOL, which is a really great outcome for all of us, I think. We have an open data approach. We've established a portal called the Kew Tree of Life Explorer, and we aim to release all of our data within 12 months of generation. You can already see a beta version of this portal at the URL at the bottom of the slide, but later this autumn we anticipate having a full launch, and through that portal you'll be able to explore the tree of life devised from our data, browse the species, browse the samples, browse the genes, and download the data and all the data intermediates all the way through to the trees. So please go and take a look at that. I think that's all I have time for, so I just want to thank the PAFTOL team, past and present, and our collaborator network, especially Norm Wickett, Matt Johnson, and Elliot Gardner. I can't thank the 1KPP project. I can't thank them enough. Without their foundation data set, we wouldn't have been able to get this far. I have many Kew colleagues who have supported us, and I want to say a special thanks to Kathy Willis, the past head of science at Kew, who really inspired this project in the first place. Thank you.
Thank you, Bill. That was a very excellent talk, and one I know which is close to both of your hearts. So I have one question for you, and then we probably have to move on. How do you think the concept of a tree of life will stand up to the increasing flood of more data from ever more species?
Thanks, Paul. I think that the methodologies we've been used to for unpicking the tree of life based on the Sanger sequencing data has given us only a small snapshot onto the processes that led to the origins of that genetic diversity. And as we increase to huge volumes of different genes being included in the tree of life, we're discovering the complexity of gene evolution, and that is causing us much greater challenges in tree of life reconstruction than we were used to in the past. So that's really the major challenge, reconciling conflicting gene history, incomplete lineage sorting, duplications, all of the stuff that genome biologists have known about for years and that perhaps phylogeneticists have ignored rather. So it's a time for a step change methodologically.
Thanks, Bill. That was a really good talk. So now we're going to move on to our next speaker, who is Emily Roycroft, who is a postdoctoral fellow at the Australia National University.
Hello, my name is Emily Roycroft, and I'm going to be talking today in this session about global resources, on some of the work that we've been doing to use the global resources that are museum collections to try to understand some of the patterns of historical extinction in Australian species. And the focus of my talk is going to be about Australian rodents. So Australia has some of the most unique mammalian diversity in the world. As well as the charismatic marsupials, we also have many species of native bats and of native rodents. But Australia also has the worst rate of recent mammalian extinction in the world. So where 1.5% of the world's mammal species have become extinct, 11% of Australia's mammals have become extinct in the last 250 odd years. And native Australian rodents have been disproportionately affected by this recent extinction. So you can see here that 19% of Australian mammals are rodents, but 47% of recently extinct Australian mammals are rodents. So there's something about native rodents that makes them particularly susceptible to the impacts since European arrival in Australia. So here are some of those extinct species here. I won't go through them all, but their names are listed along the side of the slide, and there are some pictures here of their specimens and some drawings. So while these species have been lost to us alive, many of them were collected prior to their extinction and lodged as specimens in museum collections. So that really links to museum collections as a global resource. So we have this potential resource of specimens of extinct species that we can use to try to study something about them that we didn't know. So as well as studying the physical specimens, we also can potentially use these museum collections as a really rich genomic resource. And as I'm sure many of you know, new technologies now allow us to obtain DNA sequence data from these really old specimens. And that could potentially unlock information about these extinct species that we couldn't otherwise have known and shed some light on the biodiversity that we've lost. So one of the key unresolved questions that we wanted to get at when looking at genomics of museum specimens and of extinct Australian species is whether extinct Australian species, and in particular extinct Australian rodents, were already in decline prior to 1788, or whether they went from relatively stable populations to completely extinct in less than 200 years. So the significance of 1788 is that it's when Europeans colonised Australia. And so this brought with it a whole host of different threatening processes for native Australian wildlife. And it's unclear whether these species were potentially already in decline prior to this, or their extinction was as a direct result of these threatening processes that were introduced at that time. And there's some evidence from other charismatic species like the thylacine and the Tasmanian devil that there may have been longer-term Holocene declines that predated that arrival of Europeans into Australia that might have sort of predisposed species to have become rapidly extinct as a result of that. But what about native Australian rodents? Well, we don't know much about the extent or the pace of their decline, but we can use genomic data from museum specimens to estimate what their relative population size might have been like at the time that the specimens were collected. So what I'm showing here is the average heterozygosity calculated using genomic data, in this case whole exome data, from all species of native Australian rodents. And in the grey points are species that are currently living species that are in large, relatively stable populations. And in the gold dots are living species that are in small or geographically restricted populations. And some of these are critically endangered species. And the red dots that you can see there are the extinct species. So what you can see is that extinct species are falling well within that distribution of grey dots. And these grey dots are those large or geographically connected and relatively stable populations. What this tells us is that at the time that these specimens were collected of the extinct species, which the dates are indicated next to the dots, these species were potentially relatively stable and relatively large populations. So they didn't exhibit this pattern of critically low genetic diversity that we see in those gold dots and that we might expect from a species that's in decline or endangered. And that tells us that their decline likely does not predate the recent impacts of Europeans in Australia. And that really highlights how these populations have gone from relatively stable and completely collapsed in less than 200 years. And another brief anecdote from this data that I want to share, if I have time, is that we can also use genomic data to reconstruct the phylogenetic relationships of both extinct and living species. So you can see that here, but I want to highlight one brief little example from these data. So here you can see the species level and intra-species divergences plotted as a distribution where the white points are within species and the grey points are between species divergences. And the red points are extinct species. So you can see that one of these extinct species is falling within this distribution of white points. So that tells us that this extinct species is actually the same species as a living species. So what is that species? Well, it's the Gould's mouse. And what these data are telling us is that the Gould's mouse, Pseudomys gouldi, is genetically indistinct from its closest relative, Pseudomys fieldi, the Shark Bay mouse. So what this result says is that the extinct Gould's mouse is actually not extinct. It's the same as the Shark Bay mouse, which persists on an island off the coast of Western Australia in Shark Bay. So this is just a little anecdote about how data from these extinct species can potentially shed light on the relationships between extinct and living species and inform conservation into the future. So this really highlights the importance of that persisting species in Shark Bay. So I'll wrap it up there and say thank you to my co-authors on this project that are pictured here. And thank you to the AusMammals Genomics Consortium who funded much of this work, as well as all the other funding organisations that are listed on this slide. Thank you so much for listening.
Thanks, Emily. So unfortunately, Emily was not able to join us today due to the time differences in Australia. So no questions for her at this time. But if you do have any questions, feel free to email myself and I will try to get them to Emily for her to answer. Our next speaker is Santiago Ron, who is a principal professor and curator of amphibians at Pontificia Universidad Católica del Ecuador.
Hello, my name is Santiago Ron. I am professor at Pontificia Universidad Católica del Ecuador, and I am going to talk to you about the importance of genome banks to boost biodiversity research in mega-diverse countries. This is going to be a case study in Ecuador. As you know, the biodiversity is unevenly distributed around the world. Most of the biodiversity is located in tropical countries, but unfortunately, most genome banks are located in the temperate regions, as it can be seen in this map of showing the membership to the Global Genome Biodiversity Network by genome banks around the world. As you can see here, there are very few genome banks located in the neotropical region, Africa and Southeast Asia, which are the global hotspots of biodiversity. Now, our genome bank is an exception to that rule because despite being in a tropical country, it's a large bank. We have been core members of the Global Genome Biodiversity Network for several years. Our genome bank started in 1998, and currently we have over 63,000 genome samples of animals, fungi, and plants. The diversity of our bank includes over 900 described species, but there are many hundreds of species that are undescribed and are part of this bank. Our animal genome bank is the largest in Ecuador and also the largest, one of the largest in Latin America. We keep our samples in ultra freezers at -80 degrees. And in these ultra freezers, every tube containing the samples have an individual label. And all the data from these samples is entered in a relational database available online at this address. All the data of these genome samples is also linked to the information from vouchers at the collections at Universidad Católica. We have a large herbarium collection and also fungi and zoological collections. The importance of our genome bank is that it's also linked with a very good molecular laboratory where we are constantly generating DNA sequences, which are actively being used in research. This can be easily demonstrated by conducting a search at the genome bank of the CUCAS acronym, which is the acronym for our zoological collection. And quickly, you will get over 4,700 accessions at GenBank, which is a demonstration of the large number of studies that have been using our genome's resources during the last years. What is most important is that this genome bank has been used to generate publications in peer-reviewed journals by local scientists. In Ecuador, historically, most of the taxonomic research and also the research in ecology and evolution has been carried out by foreign scientists. But during the last couple of decades, especially thanks to the development of genome banks like this and also of molecular laboratories, more and more local scientists are starting to do and carry out scientific research in taxonomy, evolution, and ecology. Thanks to our genome bank and the hard work of dozens of local scientists, we have generated since 2009 over 120 peer-reviewed publications describing over 140 species of animals, plants, and fungi. Most publications were led by local scientists, and what is most important is that the genetic information that we are obtaining from our genome bank is essential to understand the true level of biological diversity that we have in Ecuador, a mega diverse country. Some of the species that we describe end up in the popular press because they have very unique and distinct characteristics, as for example, this species of tree frog that we discovered a few years ago that was featured in National Geographic and has a very strange characteristic, which is the presence of this spine at the base of the thumb, which is very unusual among amphibians. We are also collaborating with foreign scientists from all over the world. And as an example, you can see this publication that just came out recently in Systematic Biology, where we use phylogenomic DNA sequences to present an amphibian tree of life. This collaboration was based on, in part, on genome samples obtained from our bank. As I mentioned before, the genome bank is very important to understand the true level of diversity that mega diverse countries have. Most of this taxonomic work in the past have been made using only morphological evidence. Right now we are starting to use genetic evidence, and those new data are allowing us to discover an impressive amount of cryptic species, of cryptic diversity, which in the past has not been discovered because morphological information by itself was not enough to correctly delimit the limits among species. As an example, I can show you this publication that just came out last year, on which we are describing 11 new species of frogs. Describing 11 new species of frogs on a single publication is something really unusual, considering that vertebrates in general have been subject of taxonomic work for many decades. But in this case, thanks to the use of genetic information, we were able to discover these 11 new species. And what is most remarkable is that in the clade of frogs that we were analyzing, there were only 10 described species. So in this taxonomic review using genetic data, we were able to duplicate the number of known species within this clade. And this is a pattern that we are seeing again and again whenever we use the genetic information stored in these genome banks. We are very frequently finding that the true number of species in each of these groups frequently is twice the number of recognized species now, which gives you an idea of the importance of having these genomic resources in mega diverse countries, because they are allowing to have a completely new understanding of the true levels of diversity in these tropical regions of Earth. At the end, I want to thank Pontificia Universidad Católica del Ecuador and also CENESID for providing funding for our genome bank and also the research that we are carrying out with it. Thanks.
Thanks, Santiago. That was a great talk. I have one question for you here, and that is, what are the thresholds of genetic diversity you use to delineate new frog species?
Well, actually, we didn't use a single genetic distance threshold because that is insufficient to be able to correctly delimit the species. The threshold varies depending on the processes that were responsible of generating those species. So what we use is an integrative approach. We usually combined ecological, bioacoustic, morphological, and genetic data in order to delimit those species. So basically, we try to see covariation between independent sets of characters, and based on that, we decide whether a species is new or not.
Wonderful. So now we are going to move on to our next speaker, who is Logan Kistler, who is curator of archaeobotany and archaeogenomics at the National Museum of Natural History at the Smithsonian Institution.
Hi, good afternoon from just outside Washington, D.C. I'm Logan Kistler. I'm an anthropology curator at the Smithsonian. Because this session is about global resources, I wanted to really highlight how natural history collections can be one such resource to study the evolution of our food system. So I'm going to give you a brief look at my work, where I focus on using archaeological collections and other resources such as germplasm banks, herbarium materials, and indigenous and traditional cropland races to study the domestication of plants. That's to say, the evolution of our crops from their wild relatives. Domestication is the most transformative process in human history. And that's a bold statement, but it's one that I'm prepared to stand by as an anthropologist. The reason it's been so pivotal is that almost every calorie we consume today around the world comes from domesticated species. Each of these domesticated species lives in symbiosis with humans. The plants benefit from a protected growing environment and high reproductive success because humans save seed and enrich the soil. Humans get a predictable and reliable source of food, which can often be stored for lean months in the year. These symbiotic relationships have emerged independently all over the world, beginning in the early Holocene. We see peanuts, pineapples, and cassava coming from the Amazon region, maize and squash from Mesoamerica, sunflowers from Eastern North America, and then a huge range of cereals and legumes from the Near East and Eurasia, plus sorghum from Africa, taro and bananas from Southeast Asia and the South Pacific, citrus from East Asia, and the list just goes on. And each one of these species has a unique backstory linking the plant's particular evolutionary ecology and the cultural context in which domestication took place. So why does that backstory matter? Domestication evolved over millennia as plants slowly adapted to the landscapes created by the activities of humans. So we can see this playing out plainly using archaeobotanical collections. For one example, most crops lose their natural ability to disperse their seeds during domestication. This behavior ensures a productive yield for humans because the plants hold on to their grains until the harvest. In cereal crops, this trait leaves a rough scar at the base of seeds where they had to be broken off by threshing. And it's possible to track the frequency of that trait through time using the archaeological record. Doing so reveals a situation where even the most basic domestication trait, this loss of shattering, increases slowly in frequency, taking thousands of years to reach fixation, as we're seeing here for rice, barley, and some other species. And because domestication is a symbiotic process, it only exists when it benefits both parties, human and plant. Otherwise, natural selection begins to drive the mutualistic partners into new, different evolutionary trajectories. But we rely completely on a fairly small number of these mutualisms. Over 90% of our calories come from just 15 plant species, and just 3, maize, wheat, and rice, provide over 2/3 of our calories, according to the FAO. And so in a moment with unprecedented land use change and climate instability, it's incredibly important, but not at all a foregone conclusion, that these symbioses stay robust. Let me make clear that studying domestication is not going to solve climate change or a food crisis, but understanding the long-term evolution and range of biodiversity in our food species does arm us with information that we might find useful as these challenges intensify, which I think is always a worthwhile pursuit. So let me turn now to collections and give you just a few examples of how we can put all these pieces together and dig into these domestication processes. So I use ancient DNA and genomics alongside archaeology to reconstruct these crop origin stories and global histories. So I'm going to give you just 3 little mini vignettes for just a taste of that collections-based science, both from my work and others. I'll start with maize, which is kind of the paramount staple crop. It's produced at over 1 billion tons per year, and it has absolutely incredible biodiversity in thousands of land races. Here's just a few examples from a market in Peru. Maize was domesticated in modern-day Mexico beginning around 9,000 years ago, and archaeology can be used to track its movements around the Americas. By the time of European colonization, it was the most important crop throughout the hemisphere. But a few years ago, 5,000-year-old maize cobs from collections had their genomes sequenced, and researchers learned that they were only in a partial state of domestication. So even 4 millennia after domestication had started, many of the traits that we take for granted in modern-day corn were not yet present. This really underscores the incredibly protracted, drawn-out nature of this domestication process and gives us insights into those traits that were most important for humans to target early in domestication, even by unconscious selection. Potatoes are another staple that we know and love, but they're much more difficult to find archaeologically. They don't preserve as well. And so in these cases, herbarium specimens can really come to the rescue when we want to learn about pre-industrial crop diversity from landscapes that have been drastically reshaped in the past few centuries. In 19th century Europe, a fungal pathogen completely wiped out potato harvests for several years, with the greatest impacts leading to the Great Famine in Ireland. These massive losses were partly a biodiversity problem. There was no natural resistance to the blight among the varieties being grown at that time. So in this study, researchers in Germany sequenced dozens of potato genomes from herbarium specimens, including some collected by Charles Darwin, like the one shown here, and showed that the recovery from this blight, which was so devastating, was helped by a resurgence of older blight-resistant land races with higher proportions of ancestry from the Andes. So essentially, the reinvigoration of European potato production was based on indigenous biodiversity from the Americas. And finally, I'll leave you with one seasonal vignette from my own work. Squashes, gourds, pumpkins, these all originated from all over the Americas across at least 7 different episodes of domestication. But in the wild, they're completely inedible. They're toxic, they taste awful, and their rinds are almost impenetrable. People probably first use them for their seeds, which are less bitter, and we also have evidence of wild gourds being used as fishnet floats and containers. But nevertheless, these inedible hard-shelled gourds became incredibly widespread as domesticates. So we used ancient DNA from squash, modern mammal genomes, and paleontological deposits of the gut contents of mastodons to piece the picture together. We learned that large mammals can tolerate the bitterness and toxicity in wild squash that are so strongly affects humans. But all of the massive herbivores in the Americas went extinct in the early Holocene, and the squashes were left without their natural dispersal partners. What they did was cozy up to humans. Humans provided the perfect niche for disturbed ground, and the squashes made some evolutionary concessions by becoming palatable. So this fall, make sure that you think fondly of a mastodon while you're enjoying your pumpkin pie. Thank you very much for your time.
Thanks, Logan. That was great. So I don't see any questions from the stream over here. Paul, do you have any specific questions for Logan?
I mean, one thing which interests me is how the distribution of crop specimens in museums captures the balance of wild and semi-wild material and cultivated material. Are you sequencing from primitive agriculture, or are you sequencing what were still the wild ancestors of future crops?
That's a good question. What we're trying to do is really capture a range of diversity. Oftentimes in archaeological contexts, we are sort of by definition seeing domesticated material because we're working with the crops that were important to people, and those are domesticated plants. So that's a lot of what we're working with, but we want to complement that by certainly drawing on the wild type biodiversity. So what we might do is look to herbarium specimens, sort of all over the world to try and capture the entire range of wild diversity in a crop species and use that as a comparative baseline to then track sort of the changes in diversity through time in a particular crop species.
Great. So Paul, I think I'm going to hand it back off to you to introduce our last speaker of our session today. Thanks, Logan.
Okay. So I'd like to introduce Selina Brace. She manages the Ancient DNA Laboratory at the Natural History Museum in London. Her interest is in looking at archaeological genomics to understand evolutionary events and the reaction of evolution to ecological events. And I'll now hand over for Selina's talk.
Good afternoon. My name is Selina Brace and I'm an ancient DNA researcher from the Natural History Museum in London. And I'm going to be talking to you today about recovering DNA from museum specimens, the mystery parasite resolved. Our story starts here with the short-haired bumblebee, Bombus subterraneus. This bee was once common in the south of England. It was a wildflower meadow specialist, but probably due to habitat loss, its numbers sadly declined and it was declared extinct in the UK in 2000. After this point, the Bumblebee Conservation Trust planned to reintroduce Bombus subterraneus back into the UK using wild-caught Swedish bees. Our colleague, Mark Brown from Royal Holloway University, was charged with quarantining these bees before they were released into the Kent countryside. And good job too, because after quarantining them, he very quickly realised that many of these bees were in fact infected with a rather nasty endoparasite. Because of the particular quarantine protocols that were in place, any bee that was known to be infected had to be immediately killed. This is obviously fine, except for the fact that the larvae that were infecting the bee were at this stage too small to be able to morphologically identify what larvae had actually infected these poor bees. They knew it was some kind of Centritus parasitic wasp. This is a picture of a Centritus here. As you can see, even the adults are very, very small, only a few millimeters in length. So trying to identify a tiny larva, just from the morphology, one can imagine it was very difficult. So Mark went ahead and did some PCR from the larvae, pulled out the CO1 barcoding region, but when they blasted that on the database, they got no hits. So clearly this species had not previously been sequenced. The problem then was that the only well-identified material available that was morphologically identified to then get a barcoding genetic data from was from museum collections. It wasn't very old, it was about 30 years old, but it had what they call a complex preservation history, meaning that it wasn't in a particularly good condition. Mark therefore approached us. Us in this case, by the way, is mainly two master's students, Rose Porter and Aaron Foley. But together we use typical ADNA techniques to extract and build libraries from these, from a bunch of wasps. We then shotgun sequenced them, de novo assembled the reads, and from that we're able to recover the barcoding CO1 region of, I think it was sort of 15 maybe, different parasitic wasps, and from which we built this phylogeny. This is just the CO1 region, and yay, we were able to identify our mystery larva, and I can share with you now that it was in fact Centritus politus that had infected our poor Swedish bees. So that's a nice story. We managed to solve a problem. We identified this parasitoid, go us, but there were also a couple of other interesting points that dropped out of this data set, and I'm going to use my remaining few minutes to share that with you. Point one that I think is of interest is that of read length. So what you have on the bottom here are two graphs, and this is read length in base pairs and number of reads of those read lengths. What's interesting here is that read length is of course a reasonable proxy for fragmentation length of the DNA, and what you have in the graph on the left is our Centritus. Remember, this is a 30-year-old or 30 years dead wasp, and on the right you have some data from a human that was dead in the ground for 5,500 years. What you'll see is, although you have more of the longer fragments in the wasp, your average, on average, the fragment lengths are actually very, very similar, between 60 to 80 base pairs in both cases. So what this certainly suggests to us is that there is a large amount of fragmentation or degradation that's happening quite quickly after death, which would seem to suggest a biological reaction rather than just the chemical thermal reactions that we know occur over time. The second point of interest that dropped out of this data set is that we were able to identify a polydNA virus from the wasp. So this polydNA virus is actually integrated within the wasp's own genome, within its DNA, but it can only replicate in the calyx cells, in the ovipositor of the female wasp, where it actually generates virions. So when the wasp lays her egg into the host, in our case the poor bees, in with that egg goes these virions, and it is these virions that actually suppress the host's own immune system, allowing the larvae to develop undetected. So this really is a truly mutualistic relationship between these two. The virus can only be transmitted through the wasp, and the wasp can only reproduce with the aid of the virus. So I just thought it was just quite fun and interesting that we managed to pull this out from this data set. So to summarize, important point, biological degradation, certainly the degradation that occurs shortly after death, we believe can play an important role in DNA fragmentation. What we found is that after only 30 years, DNA from the same region, so both these things were from the UK, so from 30 years is on average as fragmented as DNA that was 5,500 years old. If you remember, between 60 to 80 base pairs on average. This certainly suggests that ancient DNA techniques can be really usefully applied to even more recent and historical samples, because of course this aids the recovery of shorter fragments. And just that final reminder at the end, I don't need reminding, but just it's nice to remember the utility of shotgun NGS data to capture other DNA, and remember it can provide some sometimes unexpected results like our polydNA virus. So thanks for listening. Thanks to the funders and collaborators, and special thanks to, of course, the people who did the work, Aaron and Rose. Thanks very much. Enjoy the rest of your evening.
Thank you very much, Selena. I can see one question for you in the chat. How much of each wasp specimen are you having to destroy in order to get out enough DNA to barcode?
Oh, very little. In this case, we were actually using a single leg from these museum wasps. And as I think I mentioned in the talk there, these wasps are actually incredibly small. I could barely see this leg. In fact, I often had to get the master's student to check it was still there. So yeah, very little amount actually. We did manage to get whole mitochondrial genomes and some nuclear genes out of that as well, not just the barcoding region.
And I think we have time for one last question in the session. How do you know that the extraction library protocol that you were using was not the cause of a similar fragment length? I think that's when you're comparing the ancient human and the more recent bee wasp.
Yeah, so we did wonder about that in case we were actually preferentially biasing towards picking up these shorter fragments. But in fact, some data using similar methods we were talking about earlier today, in fact, coming from some human samples that were very, very recent, actually produced longer fragmentation lengths. So yeah, we don't think it's actually being caused by the protocols used. But that's a very good point, good question. We thought about that too. Yeah.
We've got one last question which has come in. Was it not possible to let the larvae develop and sequence them instead of the museum specimens?
Sadly not, no. Because of the protocols, the quarantine protocols that were in place, as soon as a bee was identified as being infected, both the bee and the larvae had to be immediately destroyed. So that was part of the quarantine procedure, I'm afraid. That would be easier.
Well, thank you, Selena. Thank you to all our speakers. I think it's been a really good session. We have to clear off now to prepare the way for the next session, but I hope that everyone's enjoyed it and look forward to the rest of the meeting. Goodbye.